So, where in the world did we stop yesterday? Here, we did this, done this, we're going to do the animation today of this bacterial electron transport. So, we're going to animate this just to see. Any questions on the electron transport system? One simplification of this is basically 3 components to the electron transport. So 3 sets of proteins. The first, the one that receives the pair of electrons, is this NDH1, which is NADH dehydrogenase. It is dehydrogenating NADH. So that's the first one. The second one is this pool of quinones, just in the middle, quinone pool. And then the third is cytochrome B oxidase. And the oxidase tells you it is going on under aerobic conditions. And that is where the electrons are finally dumped to what? What's the final electron acceptor? Oxygen. So oxygen is the final electron acceptor of that pair of electrons that originates from what molecule in metabolism? What molecule did those electrons originate from? Glucose. So electrons are stripped from glucose in glycolysis and TCA cycle, carried by NADH to the membrane, where they're dumped into these 3 groups of proteins: NDH, quinone, and cytochrome deoxidase. And then we talked about what are those protons. How does the cell make use of this proton motive force? So it's only by electron flow do you get protons being pumped outside. You have to have electrons, you have to have the flow of electricity to pump those protons. And then with that proton motive force, they are used to drive the synthesis of ATP through the enzyme called ATP synthase. Yeah, it is synthesizing ATP. Okay, let's see. I think I would go ahead and show that. And remember, these are on the web. In the 3rd or 4th edition, you go to the chapter and the animations associated with those chapters. And you too can watch these videos.
An electron transport system consists of a series of electron carriers that sequentially transfer electrons to the carrier with the next higher reduction potential, that is, to a stronger electron acceptor. Electron flow begins with an initial electron donor, such as a carbohydrate, and ultimately transfers all electrons to the strongest electron acceptor, which in this example of aerobic respiration is molecular oxygen.
That's a good overview.
Glycolysis and the tricarboxylic acid cycle, organic molecules donate electrons associated with hydrogen atoms to NAD+ molecules. The resulting NADH molecules act as electron donors to the electron transport system. A typical bacterial electron transport system used in respiration includes at least 3 functional components, beginning with a complex generally called a substrate oxidoreductase. This enzyme has a number of more specific names, including NDH1 for the complex in bacteria.
NDH1 stands for NADH dehydrogenase. That's the name that we want to use.
When the electrons reduce FMN, the molecule also takes on 2 protons to balance the charge, becoming FMNH2. In its reduced form,
So I'm not covering this detail, it's just cool.
One at a time to an adjacent iron-sulfur center that is a stronger electron acceptor. The iron-sulfur center passes the electron on to the next iron-sulfur center and can then receive another electron
This is in the NDH1
In a series
complex.
of redox reactions, the electron passes from one center to the next in the NDH1 complex. The transfer of electrons releases energy that NDH1 can use to pump protons across the membrane. At the end of the chain, the electrons are transferred to a quinone, which picks up two protons from solution and is thus reduced to quinol. Quinones are generally designated Q, and quinols are designated QH2.
You trap the electrons by the
the
hydrogen.
membrane and carry reduction energy to other electron transport system components. This quinone pool represents the second of three functional components of the electron transport system. The third component of the electron transport chain is a terminal oxidase complex. The complex in this example is called more specifically stadichrome BO-quinol oxidase. Within the complex is the binding site for quinol. Also in the complex are two iron-centered heme groups and three copper atoms. The quinol transfers its electrons to the adjacent heme group and releases two protons, usually across the membrane. Next, the electrons move from the first heme group to the second. This unique complex couples the electron transfer between the two heme groups with proton pumping. The iron and copper atoms pick up molecular oxygen and hold it in place until it receives a total of 4 electrons, only 2 of which have been generated so far. Note the energy from the 2 electrons donated by NADH fuel the pumping of 8 protons across the membrane. In order to become fully reduced, molecular oxygen requires the electrons from an additional NADH molecule. In total, the 4 electrons from 2 NADH molecules result in the pumping of 16 protons across the membrane. 4 protons from the cytoplasm join the electrons and oxygen to produce 2 molecules of water. In this way, oxygen, which has the strongest affinity for electrons of any other component of the electron transport system, is the final electron acceptor.
Yep. So that last stoichiometry, 16 protons, 2 moles of oxygen, it explains, that the fact that oxygen, molecular oxygen O2, must accept 4 electrons and 2 waters are produced. But I'm not stressing the stoichiometry. Okay. Entonces, seguimos. No, not with that. Okay, so we covered this cool molecular motor, ATP synthase, on Wednesday and watched a movie on it. Any questions on ATP synthase? It's another motor driven on hydrogen, on protons. Protons are its fuel. It's kind of an interesting parallel and similarity to the flagellar motor. It is powered by protons. That's its fuel. Okay, so any questions on ATP synthase? Then we will proceed with anaerobic respiration. I hardly covered fermentation to try to give you some breadth of what happens under anaerobic conditions. So life without oxygen is one of the themes that I have when I'm covering similar topics in microbial physiology. So just to fermentation is basically glycolysis with NADH nowhere to go because there's not oxygen. And so fermentation is a way to deal with recycling NAD. And that's just a short story, but just to let you know, fermentation really is a huge, important anaerobic pathway. What we're talking about here is an even more clever solution, is go ahead and continue respiring using alternate electron acceptors. So NADH can go to the membrane. Wow, go to the membrane. That's in that very corner where nobody can see that. But I just want to give you a little bit of the big picture. Is basically in fermentation, there is no membrane-bound electron transport, no ATP synthase, and so the big difference is, how much ATP is produced in fermentation versus anaerobic respiration. Let me just give you that. Gosh. Study session room, Foster Hall, 231, 1430. But here's just a quick summary of aerobic respiration gives about 38 ATP. Fermentation without oxygen and just fermentation gives a measly 2 ATP. Anaerobic, just to give you the reason why organisms mess with anaerobic respiration, about 30 ATP. So when that NADH cannot go to the membrane and dump electrons into the membrane and do electron transport, you miss all of that ATP that's produced in respiration. And so there's this selection for organisms that can use alternate electron acceptors and then continue making proton motive force and using ATP synthase, but without oxygen. And there's a big payoff. More than 10 times more ATP is produced anaerobically with respiration than anaerobically with fermentation. So just want to give fermentation a fair shake. And it's hugely successful. Organisms just run through a lot of substrate, making 2 ATP at a time, and very successful. Look at Saccharomyces yeast, which is very successful organism just living on fermentation. Okay, so what are these alternate electron acceptors? How about breathing nitrate instead of oxygen? So nitrate, this is NO3 minus, that's the formula for nitrate, can be respired. So in other words, electrons are dumped onto nitrate and then onto nitrite and nitric oxide, nitrous oxide, and N2. I cover this pathway in more detail in a physiology class. It's called denitrification. But the only point here is nitrate is a good alternate electron acceptor to oxygen. But so can chlorinated or halogenated, very toxic compounds serve as alternate electron acceptors. And so we use these abilities in bioremediating these pollutants out in the environment. And I'll talk a little bit more about that in the last couple slides. So there's a huge array of compounds that organisms can use alternatively to oxygen to dump electrons as their final electron acceptor. So nitrate, carbon tetrachloride, sulfate, iron are 4 alternate electron acceptors where you can run respiration, run electron transport, and those are the final electron acceptors. Iron, nitrate, carbon tetrachloride, sulfate. Those are 4 examples. Okay, I trot this back out because you now have a little bit more context to understand how microbial fuel cells work. And in your chapter, there is a section, in this chapter, in chapter 14, there's a section on microbial fuel cells. And so, basically, we have a microbial fuel cell here that uses the glucose and cellulose as the electron donor that is in the manure. And so in the manure are the organisms that can oxidize glucose, in other words, strip glucose of those electrons, and then carry out respiration, and we just interrupt that electron flow. So I'm just giving you this again to fill in some of the metabolic details that you now know. So here's just a little bit of a review of what I just talked about. Bacteria can respire the most bizarre of substances, mostly metals like iron and manganese, sulfate, but also these chlorinated solvents like carbon tetrachloride. And there's a huge array of halogenated compounds. My first paper that I published here at New Mexico State was on how groundwater bacteria can use the freon molecules, which are fluorinated compounds, as electron acceptors and respire with freon molecules. So breathing freon, all of these compounds that will just kill us in about a minute and a half, they are making a living off of. So organisms like this can be used in bioremediation to clean up carbon tetrachloride. CT stands for carbon tetrachloride, solid spills. But a lot of other pollutants in the environment can be cleaned up through bioremediation by stimulating this halo respiration. They are respiring halogenated groups, whether it's fluorinated, chlorinated, brominated, they like them. Okay, so that's one section. You could put a little line here, because another application of putting respiration to work is you get them to dump electrons onto electrodes and make a biological source of electricity. And so this is what we covered in chapter 1. But like I say, just in a little bit more detail, I think you have this on your outline. I've added a fair amount of detail in both these slides. But bacteria naturally present in the manure use the electron donors that also are present in the manure. So I give you the example of cellulose, which you know can be broken down to glucose. And once it's at glucose, what are they doing? They're going through glycolysis, TCA cycle, and respiration, just like we were studying. And we interrupt this electron flow for like a millisecond. They dump electrons onto our membrane electrode, and there's actually the membrane electrode is right there, and where the wires are hooked to. And then we use this electricity to run, in our case, a voltmeter, and you can run any kind of device. Flashlights, you can run small, and they're basically small devices because it doesn't generate a lot of electricity. But you put a lot of these in series and you can really amp it up. Okay, so here is more information. Do you have this slide? You say you do? Okay. So I just added more text. Let me bring it all in to this last night to expand on this and to explain. So microbial fuel cells is a topic in your chapter, the special research topic 14.2 on page 562. Well, we've done all of that work that's shown in that diagram, even before that guy published his paper on this. So let's talk about what we've been doing here at MSU with microbial fuel cells. And all we do, here is what we are learning in chapter 14. Here's a bacterial cell. Electrons are stripped from glucose, carried to the membrane by NADH, and dumped onto the membrane, where ultimately it goes to oxygen, which is then reduced to water. So That's what it says here. Normal electron flow and respiration, membrane proteins, and then the oxygen. So I'm just saying, right? This is what we've been covering for the last 2 days, and what I've been going on about it. Then here is a fuel cell, which is using the same process. So in a fuel cell, as in basically our fuel cell in what we're calling the manure battery, bacteria donate electrons to this electrode, and the electrons flow through whatever gadget you have. We have a voltmeter. The guy on page 562 has a light bulb. And those electrons continue flowing where? Well, to the cathode where oxygen is. And so all we're doing is interrupting for, microseconds, not even that, nanoseconds, the flow of electrons to oxygen. So oxygen is the final electron acceptor in aerobic respiration. We are just capitalizing for a microsecond that flow of electrons, and those electrons flow to oxygen to reduce oxygen to water. So oxygen is still the final electron acceptor. So the point is, this is normal electron flow in biological respiration. We just have interrupted the electrons for a moment, like a nanosecond, to power whatever device. So, what's fun about working with microbes is, utilizing their natural processes to do some helpful things for us. Clean up a chlorinated solvent spill by stimulating this halo respiration, or capitalizing on normal electron flow in normal respiration, and using that electricity for a little bit. And so it's just an interesting, microbial fuel cells have been developed quite a bit in the last 10 years. So a lot of work done. And one other point that I don't cover in this class, but there are structures related to what pili look like and fimbriae look like, that are extensions from the cell membrane, that are called nanowires that connect one cell to another, and they're just exactly like an insulated copper wire, where electricity is shared between bacteria and then extended out into the environment through these biological structures that are called nanowires, which are basically wires that are at the nanometer level. So a lot of interesting, fun research to be done in bioenergy, capitalizing on biological sources for energy, which is one of the things that we do in my laboratory, and as well across campus in engineering. These collaborations with Dr. Condon and Dr. Dang have been just great. Okay, so do you see the connection here in this kind of biotech application to normal respiration? So it kind of helps justify studying the basic processes when you can see kind of an immediate application. Hey, let's make some electricity. Any questions on that? Okay. So the last little section is just another type of metabolism that, instead of using glucose, how about we just use rocks? How about we use nothing organic, but something inorganic as an energy source? So, please kind of expand some appreciation for what bacteria can do metabolically. They're extremely diverse in their metabolic abilities. So lithotrophy, litho means rock, trophy means grow, so this means growth on rocks. And so it's the acquisition of energy by the oxidation of inorganic electron donors. So one type of lithotrophy that's important is this nitrogen oxidation. I'm going to just leave it at that. I go into a lot more detail in other courses. And so here is your source of energy. Ammonia. Ammonium has, you know, you have to have a source of electrons. Ammonium is a pretty reasonable source of electrons for these organisms that can carry out lithotrophy. And so I'm not really going to require this pathway for you. I don't know what they're saying. Nitric acid is nitrate. I guess these are alternatives. Nitrous acid and nitrite. Ammonium can be reduced to nitrite and nitrate as well as nitrous acid and nitric acid. So then we're going to finish up with, this is a pathway right here using ammonium and oxidizing it to nitrate that was discovered about 100 years ago by Sergei Winogradsky, and maybe even Weisring, but a long time ago. But recently, they found another pathway that actually ammonium can also yield energy under anaerobic conditions through oxidation by nitrite produced from nitrate respiration. So see next page for the Anamox reaction. So Anamox stands for anaerobic ammonium oxidation. You don't need to know that because we cover it in these other courses. This is sort of a teaser for the microbial physiology class that I teach, Biology 451, every other spring. I'm teaching it this spring, and so it's every 2 years. Okay, so the reaction that we're talking about is under anaerobic condition, nitrite plus ammonium goes through this incredible intermediate. So this is an intermediate called hydrazine, and then the product is nitrogen gas. So though I'm making exclamations about what hydrazine is, understand this is simply saying these are the substrates, this is the intermediate, and this is the product in kind of a typical enzyme reaction. And so the point here is we have hydrazine, which is N2H4, and I'm not really going after the details here in terms of alternative electron transport, but just the very basics of you've got, we want to find here is one substrate. One substrate right here is ammonium, and that is combined with nitrite. There are your two substrates. And the intermediate that's produced in this reaction pathway is hydrazine, which is what NASA has been used for rocket fuel since the mid-60s. So it's a highly explosive, pretty darn toxic compound that is so interesting to see biology can handle rocket fuel. And so then the question is, how can biology handle safely something so explosive and so energetic and so volatile as rocket fuel? And the answer to that is on the left. And so this is a multi-layered membrane called the anamoxisome. And it takes up, so here on the outside is a normal cell membrane of two cells that are actually about to split, to divide. But taking up like 90% of the cytoplasm is this massive vacuole that is multidimensional. So there's many, many, many layers here, and many, cristae, these invaginations of that membrane. So a massive cytoplasmic vacuole that is multi-layered is where the bacterium handles with safety, and kind of like an OSHA-inspected location, that, yeah, you can handle rocket fuel as long as it's in this anamoxisome, which is so thick. And why is it so thick? It's to sequester or, keep compartmentalized away from the rest of the cell activity in this anamoxisome to sequester this toxic rocket fuel from the cell. So there's a lot of interesting things about this reaction that I cover in microbial physiology. But, understand what's the substrate, the intermediate being hydrazine, and the product of this anamox reaction. nitrite and ammonium go through rocket fuel and then produce, you know, a very innocuous, very non-toxic product, nitrogen gas, which we're breathing 80% nitrogen gas. So you go from a really very, very volatile compound, hydrazine, to a very innocuous product, nitrogen gas, which is extremely stable. And this is pretty close, well, it's probably stained with the computer, but it is a photomicrograph using this cryo-EM of this organism. So, okay, that is, right, what I've covered in chapter 14, and that's what is, what we're finished, and it's nice that we have a little time left. If there are any questions, yes.
You said 4 to 6:30 on one side and 4:30 to 6 on the other. So
Oh, very interesting. Wow, thank you so much. So what I meant to say, I really appreciate you straightening